Having discussed how neurons work and communicate with one another, we'll now talk about the methods that are used to understand how the brain works. And we'll begin with experimental ablation, or another word for ablation is lesion. These studies are the only kind of neuroscience study that allows inference of necessity of a structure. Inference of necessity of a structure. If you think about it, there are two kinds of causes, necessary causes and sufficient causes. Now, in the methods lectures here, I will continually refer to memory experiments and the hippocampus. Hippocampus being a brain region that's known now to be very important for memory. But let's say we want to study the hippocampus. We could create a lesion in the hippocampus and observe that the animal's memory is no longer intact. In which case we could say, well, the hippocampus must have been necessary for memory, but of course it probably is not sufficient for memory. If I just put a hippocampus on the table, you don't have memory, right? So the hippocampus by itself is not sufficient for memory, but it does need to be there in order for memory to occur. So these lesion studies allow us, that is true of the hippocampus, this is true of any brain structure. I create a lesion and I observe a deficit in behavior, then I can feel confident that that particular brain region was necessary for the behavior that has been lost. If you think about brain imaging studies or single cell study recordings of from non-human animals, those studies are correlational. They say that this brain region activates when the animal performs a particular task, but they don't say that it has to activate in order to perform that particular task. Only these lesion studies allow us to say that the brain region was necessary. So how do we create brain lesions? Well, one way is through aspiration. By the way, I won't say much about the ethics here, but I do think the ethics of this kind of research are very important for one to consider, even though I won't really discuss it here. Lesion studies, of course, are usually done in non-human animals because we certainly don't feel it's ethical to create a lesion in human animals, although some people believe it's okay to do so in non-human animals. And so one of the most common subjects in these non-human animal studies is rats. And so what you might do is take a group of rats, and for each one you're going to put them under anesthesia and perform surgery. Now, one of the ways that you may damage or remove brain tissue from, in our example, the hippocampus, would be aspiration. Here, that glass pipette comes back into our field of focus. Remember, we talked about how you can record individual neurons by inserting an electrode into a glass pipette and then inserting that glass pipette into the cell membrane. Well, you can use a much larger pipette and put a vacuum hose basically up to it, and you can just suck or aspirate the brain tissue out. Okay, so that's one way of creating a lesion. Another method is to burn the tissue. You can take a stainless steel wire, hook it up to an electrical source, and give it a radio frequency electrical pulse, which will cause the stainless steel wire to heat up. And if you apply that heated wire to the brain tissue, then it will kill that brain tissue without killing the rest of the brain. Now, these two methods are quite useful, but they have their limitations. And in one limitation is that they will not only damage your brain region of interest, they'll damage any axons that happen to be passing through that brain region. So, for instance, we're interested in the hippocampus, so we create a lesion in the hippocampus, and we observe that memory is impaired. But we don't really know for sure, were there any axons that happen to be passing through, axons that were not related to the hippocampal function, but were just kind of a highway passing through a neighborhood, so to speak, well, any of those kind of axons will also have been destroyed. And so maybe it's not that because we destroyed the hippocampus that memory impairment was observed, but maybe it's because we destroyed the axons that happen to be running through the hippocampus. So this third type of lesion technique avoids that. It spares axons that happen to be passing by. And this kind of lesion is an excitotoxic lesion. And this is a good name because right there in the name is exactly what's happening. It's toxic for a neuron to get overly excited. The neuron will, basically explode. I mean, I'm exaggerating there. It doesn't really explode, but it'll fall apart. It'll damage itself because of being overexcited. So it's toxic to become excited. Therefore, if you introduce a chemical into the hippocampus that causes it to depolarize, to get overexcited, then that will toxify the neuron. So it's an excitotoxic lesion. And again, that spares axons that happen to be passing by. Another way of doing this is creating reversible lesions. And in this case, one way to do it is to cool the brain region. Take a refrigerant, apply that refrigerant to the brain region of interest, in our example, the hippocampus, and cool it to a temperature that doesn't destroy the cells, but does make them too cold to fire action potentials. Okay, and so you can observe the change in behavior. Now, when you remove that refrigerant, the brain returns to its body temperature, and the brain region will come back online. Another way to do this is with a chemical called muscimol. And here, this muscimol stimulates GABA receptors. GABA receptors are always inhibitory. So if you inhibit GABA receptors, you're causing neurons to become inhibited. And then a third way of doing reversible lesions is through optogenetics. Here, you modify genetics, or you modify genes to be sensitive to light, optogenetics. And in so doing, you can actually have neurons produce ion channels that are light sensitive. So you can depolarize those neurons just by shining a light on them. Or alternatively, you can hyperpolarize neurons by shining a light on them. And this, if you hyperpolarize them, then this causes the brain region to become inactive, and that's functionally a lesion. And then finally, you want to have a control group that you can compare the animals to. So, for example, if we just took a group of healthy rats, ones that had not gone through surgery, then we don't know whether the difference in memory between our control group and our experimental group is because the experimental group had the lesion created to the hippocampus, or was it just simply because they went through surgery? As I mentioned, these rats will be anesthetized. Well, we know that anesthesia impairs memory. So maybe it's just the fact that they went through the surgical process that caused the memory impairment. So we don't want to compare our experimental group against a group that has had no kind of surgery. Instead, we want to take a control group and actually run them through surgery, all the same surgery that the experimental group got, except without creating the lesion. So that means the rats have received anesthesia. That means their heads have been cut open, but no brain tissue was actually destroyed. Or we could destroy brain tissue somewhere completely different than the hippocampus. Either way, we're comparing our rats in the experimental group to a control group that has sham lesions that aren't real. So there's a little bit about how lesions are created, but how do you find the brain region that you want to create a lesion in to begin with? Well, this is going to happen through stereotaxic surgery. Okay, so the animal is, of course, sedated under anesthesia, and their head is placed in a stereotaxic device. So stereotaxis just means location of an object in space. And in this case, the object is the brain. And what they'll do is basically put some bolts into the skull of the animal to hold the head steady and in place, and that puts the head into a stereotaxic space where you've got X, Y, and Z coordinates. And then you use the stereotaxic atlas to find the brain region you're interested in. This atlas is a bunch of coronal sections of the brain from rostral to caudal to the bregma. And the bregma is the matured fontanelle. So basically, if you're looking for the hippocampus, you find in your stereotaxic atlas, you find an image of the hippocampus, and that will give you the coordinates of where the hippocampus should be found in the brain of, in this case, the rat. And you basically just measure so many millimeters from the bregma as indicated by that stereotaxic atlas. So as I said, the stereotaxic apparatus holds the head in place. You get an electrode or a cannula. So it's an electrode if you're trying to do single cell recordings and put the electrode into a specific brain region, or you're using a cannula if you're going to create lesions. And the stereotaxic device has a calibrated mechanism that moves the electrode or the cannula along the X, Y, and Z axes. And so this is how researchers find the brain region of interest. However, it's not an exact or perfect procedure, and for one thing, each brain is slightly different. And so the hippocampus might not be in exactly the same location for each animal that you perform surgery on. Consequently, after you've completed your experiment and tested the animal in, say, a memory experiment, then you're going to perform histology. This involves killing or sacrificing the animal. Then you're going to fix the brain in that should say formalin, or it does say formalin. Okay, great. So formalin is going to destroy all the autolytic enzymes and keep the brain tissue from deteriorating. But now that you've got the brain fixed, you need to slice it into thin slices that can be viewed under the microscope. And to do that, you're going to freeze the brain and then slice it under a microtome. A frozen brain slices much more thinly than an unfrozen brain. Next, you may stain the brain, and you're typically going to use a stain that's taken up by cell bodies, but not by axons or dendrites. And then you're going to examine those brain slices under the electron microscope. And effectively, you're going to look slice by slice until you find the brain damage that you've created. And for each rat, if the brain damage or the lesion is in the region of interest, then you'll go ahead and use those data. But for any rat that you actually created the lesion outside the brain region of interest, you're going to discard those data. So we've talked about how to create lesions, and then we talked about the histology process whereby we verify where our lesions were actually created. But now it's time to figure out what other brain regions are working in conjunction with the brain region of interest. So again, in our example, we're talking about the hippocampus. We have created a lesion in the hippocampus. We've observed the rats lose their memory for where a food was hidden, and therefore have been able to conclude that the hippocampus was necessary for memory. But it surely is not the only brain region involved in processing memory. The hippocampus is probably working in conjunction with other brain regions to process memory. So we would like to know, where are these other brain regions? And to do this, we can use tracing methods. We can trace efferent connections. Efferent connections would be ones that are leaving the hippocampus, or we can trace the afferent connections. Those are the connections that are coming into the hippocampus. So to trace efferent connections, we would use anterograde labeling. And this usually involves collecting PHAL, a protein produced by kidney beans, actually. And so we would take these proteins, attach a fluorescent dye to them, and inject them into the hippocampus. Now, we will wait a few days for the neurons in the hippocampus to absorb these proteins and for these proteins to migrate down the axons to the axon terminals. And then at that point, after a few days, we would do the histology, kill the animal, slice the brain up, and mount the slices on to slides, and we would examine slide through slide until we came across that fluorescent dye. And let's imagine we found the fluorescent dye in the posterior parietal cortex. Well, that tells us that the hippocampus was sending efferent axons to the posterior parietal cortex. So now we know that not only is the hippocampus necessary for memory, but that it's probably working in conjunction with the posterior parietal cortex to do that memory processing. But it's likely that the posterior parietal cortex is doing something slightly different than the hippocampus is. So what do we do now? We create lesions in the posterior parietal cortex. We observe the effects of those lesions on the behavior of a new set of animals. And then we trace that brain region's efferent connections and we repeat this until we reach the motor cortex, which would be the output of the entire memory system. So that's tracing efferent neurons using anterograde labeling, but we would also like to know, well, what brain regions are sending connections to the hippocampus? What are the afferent connections to the hippocampus? In which case, instead of using anterograde labeling, we're going to use retrograde labeling. In that case, a virus is typically used because viruses, instead of being absorbed by the cell bodies in the hippocampus, they'll be absorbed by the axon terminals. And they will climb, the virus that is, will climb retrograde up the axons until they get to the cell bodies. So by injecting this labeled virus into the hippocampus, we can wait a few days and then do the histology, look slice by slice, and now wherever we find dye, we are finding a brain region that is sending connections to the hippocampus. So that's anterograde labeling, tracing efferent connections, and retrograde labeling, tracing afferent connections. So that is our discussion of lesion techniques as well as tracing techniques. Now let's talk about a completely different technique, one that we've already discussed when we discussed how the neuron functions. And this would be using microelectrodes to do single cell recordings. As we mentioned before, this requires the use of a glass pipette, a glass tube that is fine enough on one end to puncture the neuron without destroying it. And as such, that the intracellular fluid within the neuron will flow into the glass pipette, and on the other end of that glass pipette we have inserted an electrode. That intracellular fluid, which has a voltage charge to it, will cause a current in the electrode, and we just output that electrical current to a computer screen so that we can actually observe the shape and the temporal dynamics of the action potential itself. Now, this is how you do it intracellular. How do you record from the inside? But you don't actually have to puncture the neuron. You don't have to replace the glass pipette on the inside. You could instead place the glass pipette right outside the cell, in which case you're doing an extracellular recording. Recall that an action potential involves the movement of ions in and out of the cell. Therefore, you can see a voltage change not only inside the cell, but you can see that voltage change outside of the cell as well. So that is single cell recording. We can also do macroelectrode recordings, where instead of penetrating an individual cell, you're simply placing something like a metal disc or a wire on the surface of the brain and picking up the electrical activity from tens of thousands of neurons at once instead of just a single neuron. This can be used in many contexts. In medicine, it's often used to localize spike zones in epileptic patients. Spike zones, what are those? Well, epilepsy usually involves a small area of brain tissue that is epileptic. And when a seizure starts, it starts in this epileptic tissue and then spreads from there. This small area is called a spike zone. And what is fairly common these days in patients with intractable epilepsy, that is, epilepsy that can't be fixed by medication, they will actually go in and do brain surgery, and they will remove this small bit of tissue in the spike zone. But what they have to do ahead of time is, say, about 2 weeks prior to surgery, they will insert electrodes into the patient's brain and record for a couple of weeks to find out where this spike zone is. And once they find it, that's the brain tissue that they will remove. That just about does it for our discussion of invasive methods that involve creating a lesion or tracing connections or recording from individual neurons or even tens of thousands of neurons simultaneously. But before we move on to talk about brain imaging, I would like to tell you about a relatively new advent in invasive neural recordings, and that involves multiple electrode linear arrays. Now, what we discussed for recording from individual neurons was single cell recording with a microelectrode, and we can see here in the top image, we can actually see a pyramidal cell with its cell body, and we can see the electrode or the glass pipette invading the individual neuron. And off to the right, we see the spikes or the firing pattern of the neurons. But since the advent of the single cell recordings, we now have, in the last 5 or 10 years, multiple electrode linear arrays. And these are capable of penetrating individual neurons, but instead of having a single electrode inserted in them, they may have something like, I believe this example here has 16 electrodes. So in this case, we can penetrate not just one neuron, but we can penetrate many neurons. And wherever in these neurons an electrode happens to fall, we can record from that neuron. So now you can record from 16 neurons simultaneously rather than just a single neuron at once.